for you in your D2L. I'm going to try to upload every single session, maybe in 2 hours, maybe in 5 hours. It's going to be today for sure. Then, if you remember our Tuesday session, we were talking about the lymphatic system. And if you remember the lymphatic system, we're talking about the system which is like the cardiovascular system. If you remember, we have, for the cardiovascular system, we have the heart, which is the pump, and we have the blood vessels. In this case, we have no pump because the pump is going to be our skeletal muscle. It's going to be the same thing as veins. Then the lymphatic system is a collection of cells, and we have a group of vessels called lymphatic vessels. And they are assisting in circulating fluids. They are assisting in distribution of fluids. And they are really close associated with the cardiovascular system. Then, if you remember on Tuesday, we were talking about the functions and lymphatic system, and they have this system, it has three main functions. The first function is transporting excess of fluid away from the interstitial space. If you remember, guys, I was talking about interstitial space. When you have a cell and you have another cell, this space between cells is called interstitial space. Then, usually, if we have fluid here in this space and we have an excess of fluid, then the lymphatic system is going to be in charge of transporting that excess of fluid away from the interstitial space, and it's going to be redirecting this fluid into our veins. Then it's returning that fluid into the bloodstream. It's going to be draining lymph. It's going to be draining lymph. It's going to be draining lymph into our blood. Then the first function is transporting the excess of fluid away from the interstitial space and returning that fluid, which is lymph, into the bloodstream. The second function, it has to be around lipids. And if you remember, I told you we have 4 biomolecules: carbohydrates, proteins, lipids, and nucleic acids. And from the food, we can absorb carbohydrates, such as monosaccharides, glucose. We can absorb amino acids from proteins, but lipids, we cannot absorb them directly from the digestive system into the blood. Then we use the lymphatic system for that purpose. And the third function, in my opinion, is the most important one, is defending our body against diseases, mostly infections. Then we have another system embedded in this lymphatic system, but it's not a group of organs, it's a group of cells. And this one is referring to the immune system. That's the fact we have as many cells in this lymphatic system, and they are providing protection against infections and other diseases. Then again, we have today, the lesson is going to be about lymphatic system and immune system. Check with you guys, give me a second. Then we have, guys, this is the lymphatic pathways. And by the way, you're going to have for sure a question about it during the lecture exam and the lab practical exam. This sequence, this pathway should be for sure in your exam, and you have to be able to acknowledge this pathway. Then the first thing is going to be lymphatic capillaries, and you have here the capillaries. Remember, when you have capillaries, it's because you have here tissues, cells, and you're able to interchange oxygen, carbon dioxide, you're able to deliver nutrients, and you are able to pick up some waste. Then the first step is going to be capillaries. Then from here, you're able to drain these capillaries on lymphatic veins, lymphatic vessels. These are lymphatic vessels. And then we have the first organ, which is the lymph node. And remember, I told you, we have lymph nodes. They are filters for our blood. Then we're picking from here some lymph, some interstitial fluid. Then from these capillary beds, we're sending that fluid into the lymphatic vessels and then to the lymph node. These lymphatic vessel before the lymph node, this one before the lymph node, it's called as this one, it's called afferent vessel. Afferent vessel. If you remember AP1, when we have something arriving into the ganglia, it's afferent. If we have something emerging from the ganglia, it's efferent. This is the same thing. Here we have lymphatic vessels, and when we're arriving into the lymph node, this is called afferent, and this one is called efferent. This is afferent, and this is efferent. Previous to the lymph node and after the lymph node. Remember, these are lymphatic vessels. Then we have around 400 to 700 lymphatic nodes in our body, mostly in our folds regions. Then you're going to have a lot of lymph nodes. Actually, I'm going to show you now where is located these group of lymphatic nodes. We can see here an image. Mostly we have in this region, in popliteal region, inguinal region, abdominal region, thoracic region, and then your elbow, your armpit. These regions are populated by lymph nodes, your neck. Then if you have an infection, for example, if you have an infection here in your toes, you're going to have these lymph nodes and these lymph nodes working a lot. If you have an infection maybe in a finger, you're going to have these lymph nodes and then lymph nodes this here. They're working a lot. If you have an in your eye, then you're going to have these lymph nodes working a lot. Then they are filters for infection, guys. They are filters for infections. You need to know we have around 400 to 700 lymph nodes in our body. Then, important thing about lymph nodes, you need to know this pathway. Then they're going to be draining on lymphatic trunks. And these lymphatic trunks, they're going to be draining on the subclavian vein. If you remember, the subclavian vein is going to be draining on the superior vena cava. And this way, we're returning that lymph into the blood again. Then again, guys, the first step is going to be lymphatic capillaries. This is called lymphatic bed of capillaries. Then the interstitial fluid, which is the fluid between cells, is going to be picked up here in our lymphatic vessels. These are lymphatic capillaries. And we're going to use the same system as veins. They're using the same system. If you remember, I was talking about veins a few weeks ago, and we were talking about these elements. These are valves. And if you remember, any single valve is going to be to avoid backflow. Then we're using for lymphatic vessels the same valves we're using for veins. And regarding the three layers, they have the same layers that veins have. Then they have an inner layer, tunica intima, tunica media, and tunica externa, the same amount of layers we have for veins. And again, they have these valves to avoid backflow. then the ideal path is going to be capillaries, lymphatic vessels, lymph nodes. Before the lymph node is going to be afferent vessel. After the lymph node is going to be efferent vessel. And then we have lymphatic trunks. And finally, we're draining on the subclavian veins. You need to learn this path. You need to learn this sequence for sure. You're going to have this sequence in your exam. Another important thing to recall here, it's going to be on the right side, you have the right lymphatic duct. This is the great duct to drain the whole right side. But on the left side, you use the thoracic duct. Again, a huge difference, and it's going to be, again, maybe in your exam, and it's something you need to know. On the right side, you have the right lymphatic duct, but on the left side, you have the thoracic duct. This is important, guys, because this one is draining on your left side, on your left subclavian vein, and this one is draining on your right subclavian vein. And both of them, both subclavian veins, are draining on the superior vena cava. Again, this is important to know. You're going to have for sure a question about that in your lecture exam and your practical exam. Guys, this is the path, and this is the sequence you need to know. The lymphatic capillary bed, usually you have the last name here, capillary bed. Then you have the afferent lymphatic vessel before the lymph node, and after the lymph node you have the efferent lymphatic vessel. Then you have the lymphatic trunk, the collecting duct, and finally you're going to be draining in the subclavian vein. Remember, collecting duct for the right side is the right collecting duct, and on the left side is going to be the thoracic duct. You need to remember this for your exam. Tissue fluid and lymph fluid. Guys, if you remember, I told you we called the space between cells, we call this space interstitial space. This fluid here is called interstitial fluid. But when we have this fluid inside the vessel, then it's called lymph. Then first we have interstitial fluid. But then when we have that fluid inside of the vessel, it's called lymph. By the way, lymph is almost the same composition we have with plasma, but it's going to be with less proteins. That's the only difference. It's the same thing as plasma, but it's with less proteins because this fluid, this interstitial fluid, has less proteins. Again, the fluid here is called interstitial fluid between cells. But after we have that fluid inside of the lymphatic vessels, it's called lymph. And the composition is going to be almost a plasma composition, but with less proteins. Check later, guys. It's the same thing here. This is important, guys. I told you we don't have a pump like the heart to move the lymph. Then we need to move our lymph through muscle activity. It's going to be the same thing we use with muscles, with veins. Normally, from the heart to the body, we use the heart as a pump. But from the body to the heart, we only use our movements, walking movements in generally, to pump that blood from the body. It could be from the toe to your heart, from the fingers to your heart, from the head to your heart. It's going to be movements of muscle tissue, and it's usually skeletal muscle. We use the same way in order to get that that kind of movement of lymph from the body, from the interstitial cell through lymphatic vessels, filtering that lymph through the nodes, and then sending that lymph to the heart, back to the heart, using the specifically the subclavian veins. I'm gonna share with you my screen, guys. Let me know in the chat if you have so far any questions. Remember, we're talking about the lymphatic system and immune system. We're talking the same thing we were talking during the Tuesday session, but we're going to be in detail here because we need to expand this knowledge for you guys. Guys, remember to check your chapter reviews. It's going to be a good source for your lecture exam. And remember to check the Biology Learning Center for your lab practical exams. It's going to be a perfect source of information. Remember, you can use whatever you want for the lecture exam. You cannot use anything for the lab practical. I'm going to share with you my screen again. Let's see. What else do we have here? Another function, guys, I already told you. It's the absorption of fats or fatty acids or lipids. Remember, we can absorb directly carbohydrates, glucose. We can absorb directly amino acids into the uh, cardiovascular system. We can get directly into our blood proteins and carbohydrates. But lipids, it's different because they are bigger molecules. Then we need to absorb these bigger molecules using this path, using the capillaries. Then we're using these capillaries in order to drain our lipids inside of the blood. Regarding the organs, guys, you remember I told you regarding the lymphatic system, we have organs and we have some tissues associated with the lymphatic system. Then among the organs, we have lymph nodes, we have the thymus, and we have the spleen. And as tissue associated, and it's called mucosa associated lymphoid tissue or MALT, we have tonsils, appendix, and Persian patches. They are not really, they are no organs, but they are tissues embedded inside of real organs. For example, in your pharynx, in the mucosa of your pharynx, you have tonsils. And remember, you have three kind of tonsils. You have palatine tonsils, you have pharyngeal tonsils, and you have sublingual tonsils. And all of them are lymphatic nodules or lymphatic tissue in charge of defending our mucoses. And we have another important, very important portion of the lymphatic tissue associated with mucoses, which is the Persian patches. If you go over the mucoses of the small intestine, especially jejunum and ileum, I'm going to talk about that in digestive system, you're going to have a lot of lymphatic tissue embedded in that system. And then again, the organization of the lymphatic system, we have organs, specific organs with a capsule, and then some reticular connective tissue inside filled with immune cells. These are lymph nodes. Remember, lymph nodes are filters, thymus and spleen, and some other tissues. They are not organs. They are embedded in organs, such as tonsils, the appendix. We're going to talk about that in detail during the digestive system. And Persian patches, again, embedded in the small intestine. Guys, first of all, we have our lymph nodes. If you remember, I told you the lymphatic vessel arriving into the node. This one is called afferent lymphatic vessel. Afferent lymphatic vessel. If you have the lymphatic vessel departing after the lymphatic node, it's called efferent lymphatic vessel. Then you have here afferent and efferent vessels. This is the lymphatic node. Then here you have a capsule, and you have the cortical region, and you have the medullary region. The capsule is made of connective tissue, and the region, the cortical region, has many germinal centers. These germinal centers, we have here many places with lymphocytes, and we have this medullary region, this inner region. It's filled with macrophages and T cells. If you remember, monocytes, it's one type of a white blood cell, and it's called monocyte when we have those cells in the bloodstream, but they are migrating out of the blood vessels, going into tissues, and that way they are called macrophages. They are specialized in phagocytes, in engulfing and eating solid particles such as viruses, bacteria, etc. Then we have T cells, lymphocytes, lymphocytes T, as in Texas. T cells, they are in charge of the cellular immunity. We're going to be talking about that in a minute. Then, guys, we have lymphocytes and macrophages as the main cells in order to produce here immunity, in order to filter our lymph. After that, we have, I already told you the location. We have the two main functions of these nodes. Then they are filters. Again, they are filters for potentially harmful particles from the lymph. And they are as well the surveillance, immune surveillance. Then we're able to be as a vigilant of our blood because we have macrophages and lymphocytes there. Guys, the second organ I told you is an organ only for kids. We have thymus. It's well developed, around 2 years old, and it's going to be active, very active, until before puberty. And again, it's a gland located above the heart. It's located in our mediastinum. Remember, the mediastinum is the cavity between lungs. This is the mediastinum. And we have the heart. We have the place for production and maturation of T cells. T cells. Again, we produce in this gland, which is active before puberty, in our thymus, T cells. Actually, they are called T cells because of the thymus. And these glands produces these hormones. These hormones are called thymosins, and they are for the maturation of these cells. Then again, we have the thymus. Thymus is really active as a gland, active only before puberty. We have another organ, which is the third organ, and it's a little bit different. It's the spleen. Remember, it's located behind the stomach. It's located in our left hypochondriac region, but behind the stomach. And we have three functions here. We have storage for red blood cells. We have the same function as the lymph nodes. It's a filter for potential infections. And we have, this is the place for, as a graveyard. It's the last place for red blood cells. Then we have three functions. It's a filter, like the lymph nodes. It's the storage for red blood cells, and it's as well the graveyard of red blood cells. If you remember, I told you the lifespan for a red blood cells is 120 days. And after that, we're going to have the spleen as a graveyard for the red blood cells. Guys, if you consider the histology, we're going to have two types of pulp. We have the red pulp and white pulp. As you can see here, the white pulp is in here. You have here lymphocytes, white blood cells, immunity cells, and you have red pulp filled with red blood cells. And again, this is an organ useful as a storage. For example, if we have anemia, we're going to have storage of red blood cells here. If we have bleeding, we're going to have a red blood cell storage here. And it's the graveyard for old red blood cells. Then after 120 days, they're going to die here in our spleen. These are the functions for our spleen. Let's see what else we have here, guys. This is very useful as usual, guys. After reading your slides, after reading your material, after reading your textbook, after reading your lab manual, these charts are very, very useful, especially for the exams. Then you have here the summary. You have here the organs. We have 3 organs as the organs of the lymphatic system: lymph nodes, thymus, spleen. You have the specific location, and you have the specific function for each of them. Then, guys, besides the organs, we have the mucosa associated lymphatic tissue. And remember, we have appendix in our intestine. We have the tonsils, and we have 3 kinds. We have palatine, pharyngeal, and sublingual. And we have the most important part of the mouth, or mucosa associated lymphatic tissue. It's in our small intestine, and it's called Persian patches. Guys, the last function, and in my opinion, the most important function that we have in our lymphatic system and the immune system, which is embedded in the lymphatic system, is our defense against infections. Then here it's going to be better if we consider some concepts, such as pathogens. It's good to know. Pathogens, it's any particular disease-causing agents. It could be a bacteria, it could be a virus, it could be a parasite like a protozoa, it could be a fungi, it could be anything. But these pathogens are able to produce these diseases, these infections. Then we have 2 types of defense against these type of infections. The first level is the barrier, and remember, I told you, it's the skin, is your mucus. In your mucosas, you're going to have mucus in your respiratory system, in your digestive system, in your urinary system, in your genital or reproductive system. All of them, they have mucus to trap germs. And finally, we have the pH. The pH on your sweat, the pH on your tears, the pH on your gastric acid, which is really acid, pH 2. They're going to be the first barrier against infections. Then you have a second level, and remember these 2 words: innate and adaptive immunity. This is the immunity that we have against infections. One is non-specific and the other one is really specific. Then the innate is going to be a general defense, and it's going to be non-specific, always the same mechanism to attack an infection. And we have another one which is specific, and this one is more precise. It's targeting the specific antigens. Then it's going to be a different strategy against a bacteria. It's going to be a different strategy against viruses. It's going to be a different strategy against fungi, or it's going to be a different strategy against a parasite. Then we have two kinds and several levels of defense. The first level is going to be barriers. The second level is going to be innate immunity. And the third level or specific level is going to be the adaptive immunity. Let's see what we have here. Then some innate or non-specific defenses, guys. This one is really and it's a particular and general defense against infections. Some species are resistant to certain diseases which other species are susceptible. For example, we have infections that dogs and cats are not able to earn. And we are resistant against their infections because we have specific germs to cause diseases in our body. And they have specific germs to cause diseases in their bodies. Then I was talking about these mechanical barriers. Then it's going to be your skin and mucoses. Remember, where you have mucus, respiratory tract, digestive tract, urinary tract, or reproductive tract, you're going to be able to trap in your mucus those potential germs to cause infections. And third is going to be your pH, because you wash away some microorganisms and you attack them with their pH. It could be basic or it could be acid. Then you have a group of mechanisms, and they are innate. They are always the same. Inflammation. We were talking about inflammation. Then I told you guys, imagine you have a trauma in your head. Then you're going to have a bump here. Then that bump is going to be painful and it's going to be hot. Then you have pain and heat in the same place. And you're going to have vasodilation. Why? Because you had the trauma. And you're going to have swelling there. It's going to be a pump. It's going to be a volume, increase the volume. And you're going to have redness. Then redness, heat, swelling, and pain are the 4 signs for inflammation. And you're going to have always these signs for the same cause. It could be a virus, it could be a bacteria, it could be a parasite. Whatever you have, you're going to have the same method, and it's going to be inflammation. It's not specific. It's always the same. Then you have another general method, which is fever. And I was talking about fever. Guys, if you remember the normal temperature, it's 97 to 99.5 if we're talking about Fahrenheit, or 36.5 to 37.5 if we're talking about Celsius. Remember, fever is going to be if you have more than 101.3 for Fahrenheit, or if you have more than 38.5 if we're talking about Celsius. Then again, fever, it's a mechanism defending our body against infections. And I told you why on Tuesday. Guys, because we're able to inhibit microbial growth. Normally, they are happy to be in our temperature. Then, because we have our hypothalamus telling us we're going to have normal temperature, and our normal temperature is this one, they're going to be growing as well. Then the strategy, because they are there when we have an inflammation, we're going to be able to release some other substances called pyrogen. Pyrogens, substance. These substances are increasing our set point in our hypothalamus. Then now our normal temperature is going to be about 38.5 Celsius or about 101.3 Fahrenheit. And this is going to be a new normal temperature. And that new normal temperature is going to be to destroy or to avoid the microbial growth. We have another kind of methods as innate defenses, such as phagocytosis. Phagocytosis, if you remember, neutrophils and monocytes, when we have monocytes in our tissues, are macrophages. They're able to engulf and destroy foreign particles and cells. Again, these methods are general. These methods are non-specific. All of these are part of innate defenses against infections. Then we have, guys, species resistance, mechanical barriers. It's going to be our skin, and we have chemical barriers such as we have in our secretions, natural killer cells, inflammation, phagocytosis, fever. And now, guys, we have our third level of defense. First of all, we have our barriers. Then we have innate defense. And finally, we have immunity. We have specific defense against infections. Then we're able to design a specific strategy against a particular bacteria. We're able to design a specific strategy against a particular virus. We are able to design a specific strategy to fight against a specific fungi. Then this is a specific, and this response is based on the ability to distinguish molecules that are self, part of the body, or non-self. Then we're able to identify which is a strange cell, and we're going to try to destroy that cell. Because we cannot destroy our own cells, then we need to be able to distinguish between what is own and what is strange. Then the first concept here, useful, is going to be antigen. Antigen is going to be any particle that you have able to wake up your immune system. You're able to stimulate, to elicit an immune response. And we have two kinds of responses. We have cellular response and we have humoral response. By the way, cellular response is going to be because we have T cells. And humoral response is because we have B cells. Then humoral is because we are able to produce antibodies. And we're able to attack cells or defend cells because we have T cells. Again, an antigen is a substance of our system. It's stimulate. Then anything we have in our body, it could be own, it could be exchange, and it's going to be able to wake up our immune system. It's going to call antigen. And we have two kinds of defenses against these antigens. We have a cellular response, and it's mediated by T cells. And we have another kind of response, which is humoral. Cellular is because of cells, and humoral is because of tissues. Then T cells, lymphocytes T, as in Texas, it's cellular response. And humoral or tissular immune response is mediated by B cells, lymphocytes B, and this is the creation of antibodies. This is the cells responsible for creation of antibodies. Let's see what else we have here, guys. Then, as I told you at the beginning, guys, we have three levels. This is the first level of defense: skin, mucous membranes, pH. This is the first lines of defense. Then we have a second line of defense, which is the innate defense. This is general. This is not specific. If we're not able to stop our infection here, the germ is going to be trying this method. Then we have here Natural killer cells, inflammation, phagocytosis, fever. If a germ is able to defeat this level, then we're going to get a third level of defense. It's going to be the third line of defense. And remember, this is immunity. This is a specific. By the way, because of that, this is non-specific. This is a specific. This is faster. This is slower. Why? Because we need to design that specific method to defend our body. This has no memory, or we have no memory about infections regarding the innate defense. We're going to have always fever the same way. We're going to have always inflammation the same way. We're going to have always phagocytosis the same way. It doesn't matter which type of infection it's going to be. It doesn't matter what kind of germ it's going to be. But with the third line of defense for immunity, for acquired immunity, we have this kind of immunity is going to be with memory. Then we're able to identify any infection for the first time. Then we're going to meet that infection. Nice to meet you. I'm going to destroy you, but I'm going to be slow the first time. With the second time, we're going to be able to destroy this germ faster. Why? Because we already know this germ. We have memory about this germ. Let's see what else we have here, guys. Then an antigen could be any protein, any polysaccharide, any glycoprotein, any glycolipids. It shouldn't be a whole germ, just a small part of a germ. For example, usually when you have bacteria, they have on the surface, they have some specific proteins or glycolipids or glycoproteins. This piece of a specific material, it's enough to wake up our immune system. Then you don't need the whole bacteria to activate our immune system. Then that way, that piece of molecule is going to be called an antigen. Then remember, guys, we have lymphocytes, B cells, and T cells, and these are the main cells for immunity. Remember, T cells is for cellular immunity, B cell for humoral immunity. These are specialized in thymus. Remember, T cells, thymus. And these are released from the bone marrow. And the origin is going to be in the bursa of Fabricius. Then, guys, this is the reason they are called B lymphocytes, because of the bone marrow and bursa of Fabricius. And T cells are called T because of thymus. These are for cellular. T cells are for cellular immunity. Always you're going to have questions about this. T cells are responsible for cellular immunity, and B cells are responsible for humoral immunity. Let's see what else we have here. This is the way to get the lymphocytes. Again, B cells mature in the bone marrow, and T cells mature in the T of thymus gland. Remember, the thymus is a gland located above the heart, and it's active only when we are children. It's going to be really active until we reach the puberty. Again, when you read the whole material, after reading your slide, after reading your textbook, you have available these charts, and they are the summary for this thing. Then the origin of T cells, red bone marrow, but they're maturing in thymus. And the origin for B cells, they are maturing in the red bone marrow. That is the reason, remember, T cells, thymus, and B cells, bone marrow. We have here the location, which is general. By the way, I don't need the specific location for these guys, but you need to know all these organs, all of these mild tissues, all of them have lymphocytes B and lymphocytes T, all of them. 70 to 80% of T cells are located in blood. 20 to 30% of B cells are located in blood. Functions. Again, T cells, cellular immune response. I'm going to repeat this several times. T cells, cellular immune response. B cells, humoral immune response. They are able to kill the antigen. They are able to produce antibodies. Two different kind of immunities. What else do we have here? Let me check something more important than that. Nice. B cells and T cells, they can mature. And after that, they're going to have two kind of cells or three kind of cells. B cells, they have after maturation, because at the beginning they are naive. They are uneducated. Then after maturation, they're going to have two kind of cells. One is going to be the snitch. This one is going to, no, I'm going to kill you, because we need to remember the contact, the previous contact with the cell. And the other kind of cell is plasma cells. And these are the cells in charge of the antibody production. Then we're able to produce antibody because of the B cells, and specifically because of the plasma cells. And remember, we have as well T cells, and they can have three type of cells. And these three type of cells are T helper or CD4. We have three kinds. I'm going to tell you. We have three kind of T cells. CD4. By the way, CD4 is a protein. These are called helpers. We have CD8. And again, CD8 is another protein. And these are called cytotoxic. They are killers. And we have memory cells. Then for T cells, we have three kinds after maturation. For B cells, we have two types. We have only memory cells and plasma cells. And these are T cells after maturation, helpers because they help B cells to be mature, cytotoxic because they are able to kill directly a cell, and memory cells, they are the snitcher, they are the pointers. I know you, I'm going to kill you. On the other hand, we have B cells. And after maturation is going to be memory cells and plasma cells. Remember, plasma cells are the ones in charge of antibody production. This is really important, guys. And you need to remember this part, because we're going to be asking this in your lecture exam, in your lab practical. Actually, it's really important because it's going to be material for your HES exam, this part. If you remember the division of T cells and B cells, and you remember T cells are for cellular immunity and B cells are for humoral immunity, you're going to be fine with the HES exam questions for this part. These details about the activities for B cells and T cells, they are good for, as usual, I can tell you what things are important for exams and what things are important for life. These particular things are important for exams. Maybe in 2 weeks after the exam, you won't remember anything with that. But you need to know for life, for your career, you have two kind of lymphocytes. You have B cells. They're going to be in charge of antibody production. And you're going to have T cells, and they're in charge of cellular immunity. You have B cells, and they're going to be plasma cells and memory cells. And you have T cells, they're going to be CD4, which is a protein, remember that. They are helpers, they are helping mature the B cells. They have CD8. CD8 is another protein. Again, you have, these are the killers. These are the ones killing or eliminating those cells with antigens. And we have memory cells. And you already know the function. Guys, the last part of the session is going to be about antibodies. And we're going to talk about antibodies now. If you remember, I told you our blood is divided into elements, red blood cells, white blood cells, platelets, and the liquid part or fluid part. Then in your plasma, you have water, 92%, and 7% of your plasma is going to be proteins. And I already told you, actually, let me write it down because you need to remember this. Plasma is going to be 92% of water, and you're going to have 7% of proteins. And I told you guys, out of these 7%, you have mostly it's going to be albumin is the main component, which is the magnet albumin. And then you're going to have 36% of globulins. And among these globulins, we have alpha, beta globulins, and these two are for transporting lipids, and we have gamma in globulins. These gamma globulins are the antibodies. They are called immunoglobulins, or for shorter, it's going to be Ig. Then we have every single Ig, it has to change, light change, and heavy change. And it has a variable region because we have 5 type of Ig. 5 type of Ig. Again, they have 2 heavy chains, they have 2 light chains, and this portion at the end, is the variable region. This portion at the end is the variable region. Then guys, you need to know these 5 steps. We have IgG, which is the most common one. We have IgM, we have IgA, IgD, and IgE. Guys, IgM, they're going to be the first responder for your infections. They're going to be the faster response that we have. They're going to last among weeks. This is the first responder. Then we have IgG, which is the most common antibody that we have in our blood. 80% of your antibodies, they are IgG. Then we have IgA, and it's about 13% of the antibodies. And usually, when you have secretions such as tears, sweat, saliva, mucus, whatever you have secretions, you're going to have IgE. They are for protection of these glands. Then you have IgE. IgE, it's as well in your secretions, but it's promoting inflammation and allergic responses. Then, every single time you have an infestation because of parasites, or you have an inflammation, or you have an allergic response, you're going to have this IgE elevated. And finally, when you compare lymphocytes B and a lymphocyte T, I told you on Tuesday, normally B cells, they have an antibody, and T cells, they have a receptor. Then the marker for B cells is the IgD. It's on the surface of most of lymphocytes. It's the marker for lymphocytes B. Let's see what else we have here. This part, I think, is the most important part. Guys, another important division is about our immunity. It could be natural or it could be artificial. Again, our immunity, it could be natural or artificial. And it could be active, it could be active, like this too, or passive. Then we have natural active is going to be the exposure to our disease. For example, if you suffer chickenpox, you're going to be immune forever for your chickenpox. If you don't suffer the disease but receive the germs, because artificially you're receiving the germs, that is an example of a vaccine. Then you're able to receive the germs or part of the germ, an antigen, and you're able to activate your immune system and you're able to respond that way. This is a vaccine. This is the perfect example for a vaccine. But you can receive, instead of the antigen, you can receive the immunoglobulin. Then it's going to be artificially, but passive. And one natural way to receive antibodies is going to be when you receive antibodies from your mother. It could be through the placenta or it could be during the lactation. Then natural is going to be the disease or the contact with your mother. And artificial, when you receive the external way to receive antigens or antibodies. It's going to be active if you receive antigens and your system have to develop the immunoglobulins. And it's going to be passive if you are receiving only the antibodies. Finally, guys, we have a concept here, and I'm going to show you, and this one is the last concept I would like to show you. Is the hypersensitivity reaction. Guys, normally we have a normal response against any antigen. It could be against a bacteria, a virus, a parasite, or fungi, whatever we have. But against that infection or infestation, it could be an exaggerated reaction. Then we have an exaggerated immune response. This is called hypersensitivity, and we have 4 types of hypersensitivities. It's going to be important if you can identify, especially these 2 types, the one which is caused by immune complex reaction, and it's going to be autoimmunity. We're going to be able to attack our own cells. This is type 3. And the other one, which is really, really important, is a type 2. For example, when you have mismatched blood transfusion, then you're going to have agglutination because of a mismatched blood transfusion, and then that is type 2 hypersensitivity. Guys, this is going to be the end of the session. I'm going to stop sharing with you my session here. Could you tell me, guys, if you have any questions, any concerns, any doubts about the homework? I'm going to be uploading your learning outcomes tonight. I'm going to be uploading the learning outcomes for your homework tonight. And you're going to be able to check these material tonight. And again, guys, please check your slides. Please check your chapter reviews for the lecture exam. Please check your biology learning center for the lab practical exams. I would like to increase your knowledge besides your grades, obviously. I would like to help you guys. You need to help me checking these things. I'm going to be uploading this session tonight. It could be in 2 hours, it could be in 3 hours, it could be in 5 hours. It's going to be today. And see you on Tuesday if you don't have questions or concerns. I'm going to give you 30 more seconds, guys. I'm going to stop recording now. Stop.